Hey, hi guys, here we are. It is 5 o'clock. It is Tuesday, and therefore we have another little biology lesson for you. This is gene therapy this afternoon, and I'm really hoping that this isn't going to take us too long. This could be quite short and sweet. So, having had a look at all the syllabuses for this topic, the ones where it comes up that I have been able to find it, AQA, OCRA, OCRB, and EDUCAS. That's the WJEC syllabus for English students. I'm absolutely convinced that it's the same for Welsh students as well. So those are the four boards that I could find this topic that was included in the syllabus. Please do, as always, check with your specification and check with your teachers. I am confident about the OCRA and the AQA aspects of this, just to kind of put that out there. Okay, so we are going to go through what gene therapy is, what it's used for. We're going to talk about, for those lovely EDUCAS students, we're going to talk a little bit about muscular dystrophy. And for those OCRB students, we're going to talk a little bit about cystic fibrosis and severe combined immunodeficiency. SCID. We're going to talk about how gene therapy actually happens. And then we're going to talk about the difference between somatic and germline gene therapy and what their significant differences are. And finally, we're going to talk about a few advantages and disadvantages because, of course, this is a topic where the ethics of it is discussed. So, little bit of context first of all. Now, not surprisingly, to understand gene therapy, you need to understand genes. So really quickly, I'm going to remind you that genes code for proteins, and those proteins give rise to certain characteristics in organisms. So, for example, we could be talking about petal color in flowers, fur length in polar bears, or whether somebody has or doesn't have an enzyme working in their body, or indeed how active that enzyme is. Now, then we come on to alleles, and alleles are different forms of a gene, and they code for variants of those proteins. And therefore, they cause variants of those characteristics. So we might have a variety of petal colors, we might have a variety of fur lengths, and we might have a variety of activities in the enzyme action, or we might have no enzyme action at all, for example. So, and then this is the thing that I need to point out to you, and this is important for understanding why recessive alleles are so important in terms of some of these genetic disorders. Because practically every recessive allele codes for a non-functioning protein. Now, hopefully, therefore, it makes sense that if we think about the fact that chromosomes come in pairs and therefore genes come in pairs, one from mum and one from dad, you therefore have two copies of each gene, both of which get expressed and get turned into, they code for proteins and get turned into proteins. So does it make sense that both homozygous dominant individuals and heterozygotes both have at least one allele that is going to be able to code for a functioning protein? Homozygous dominant individuals and heterozygous individuals have the working protein being made, and therefore they display the dominant characteristic because the protein's working. Whereas if somebody is a homozygous recessive individual, that means very likely that a protein is not being made or it is non-functioning. No, in fact, I should say that again. It means that a non-functioning protein is being made. And therefore, that brings about a different variant of the characteristic, often that nothing is happening. So, for example, if there was no color proteins being produced, it might well mean that the petals ended up being white, not a particular color. If, for example, that both enzyme proteins were recessive, quite possibly the enzyme wouldn't be working. And therefore, there might be particular symptoms shown in the individual because that protein might well be crucial to their normal functioning of their body. And that's what we're going to talk about here. Because genetic disorders or genetic diseases, depending on the severity of them, can be caused by both dominant and recessive alleles. But I'm going to say at the outset that it's the recessive alleles that cause, that I'm going to say that your A-level syllabuses generally focus on disorders caused by recessive alleles. So let's talk a little bit about gene therapy, what it is and how it works. Well, this is obviously medical treatment. It's medical treatment for humans. So we're not talking about flowers or polar bears. We're talking about humans. It's a medical treatment for a genetic disorder or a genetic disease that alters or replaces the alleles that cause the disorder. And nearly always, it's the recessive allele being replaced with a dominant, or one of the recessive alleles being replaced with the dominant one. So if the disorder is caused by a recessive allele, a dominant allele is added. And this is the case in muscular dystrophy, cystic fibrosis, and SCID. All of these are recessive diseases, recessive genetic diseases. So this is what the genotype looks like. The individual has inherited a recessive allele from both their father and their mother. So, but if a dominant allele causes the disease, then often an extra bit of DNA is inserted into the allele in order to make it inactive. In other words, you're almost changing the dominant allele into a recessive allele because the functioning protein, cannot be made because there's this extra bit of DNA inside the allele. Examples of dominant allele diseases, genetic diseases, are Huntington's or achondroplasia. And that is a form of dwarfism. Just notice these are not yet treated by gene therapy. It's why I'm putting the ones that we talk about that are treated by gene therapy in black. These aren't treated by gene therapy, but they are examples of dominant genetic diseases. So for the OCRB and the EDUCAS students, let's have a quick summary of these disorders. So muscular dystrophy. Now, the most common genetic inherited form of muscular dystrophy is Duchenne's muscular dystrophy. And this is a recessive condition where none of the protein dystrophin is made. And dystrophin actually links those myofibrils in the muscle fiber to the sarcolemma or the plasma membrane of the muscle fiber. And therefore, this protein confers strength on a muscle fiber. So if it is absent, it means that the muscle fiber gets weaker. It gets weaker and weaker. And just for the record, this is a sex-linked disorder. So you can easily see how exam boards might well put together your inheritance topic and this topic in a question. Cystic fibrosis is again a recessive condition where chloride ion channels in both the lungs and intestinal epithelial cells are non-functioning. They do not allow chloride ions to pass through them. And that has a huge effect on osmosis. And it ends up making the mucus that naturally lines our lungs or our airways, our bronchioles and our trachea, and that lines our intestines, it makes that really thick and sticky when it should be much more watery and much less sticky. Now, the issue is the thick and sticky mucus in the trachea and in the bronchioles causes the cilia to be unable to function, and therefore the mucus can't be wafted up the trachea and got rid of. And as a result, this hugely decreases lung function and it hugely decreases digestion. And that means that individuals suffering from cystic fibrosis have the kinds of symptoms that they do. They struggle with their lung function, with clearing the mucus from their lungs, and therefore will have trouble breathing adequately, particularly on exercise. And their digestion is nothing like it is in a normal person or in a healthy person. And therefore, particularly as children, they might struggle to put on weight, for example. Just for the record, it's non-sex linked. And finally, we come to SCID, severe combined immunodeficiency. Now, there are several types which involve several mutations of different genes, but the two most common types cause a defective receptor on the T and B cells, and it means that those T and B cells don't develop in the fetus. The baby is born without those T and B cells, and therefore without a functioning immune system. This is sex-linked, so it's going to affect, this type is going to affect boys than it will, more boys than it will girls, just like the Duchenne muscular dystrophy. And another very common type is where a non-functioning enzyme is made, and that means that the T and B cells that are there can't undergo that clonal expansion, which is a normal part of your immune response. And therefore, those individuals have a non-functioning immune system. Now, I don't think you need to know all of the details, particularly with regards to SCID. I think cystic fibrosis, most of this you will have covered at GCSE. Muscular dystrophy, you don't need to know very much more than what I've given you here, but at least it gives you an idea as to how these diseases affect the individuals that have them. So now let's talk about the difference between somatic gene therapy and germline gene therapy. Well, somatic gene therapy affects or changes the alleles that are in specific somatic cells. And soma means body. So this means body cells, what I would call your normal body cells. In fact, any cell other than your gametes. In other words, all the diploid cells in your body. Now, as a result of this affecting somatic cells, depending on where the genes are injected or given to the body, means that only specific tissues, means that only specific tissues of the patient are affected. So, for example, let's just remind ourselves what vector means. Remember that vector, you'll have come across this in your genetic engineering topic, where we change the genetic makeup of an organism by putting a gene from one organism into another organism. And that happens as a result of using a vector to transport the gene from one organism into another. So the vector is the delivery mechanism for the desired allele to get into the cells of interest. And the cells of interest, not surprisingly, change depending on the genetic therapy that we're dealing with, depending on the genetic disease that we're dealing with. So for muscular dystrophy, the vector is injected into muscles. For cystic fibrosis, for one type of cystic or one treatment, one gene therapy for cystic fibrosis, the vector is inhaled into the respiratory system, just like asthma medication is inhaled into the respiratory system. And for SCID, the vectors are introduced into the blood stem cells or into where there's a bone marrow transplant, that those functioning T and B cells can be produced. So this only affects the specific tissues of the individual affected by the disease. Now, germline gene therapy is quite different because, importantly, this alters the alleles in the gametes, in the sperm and in the eggs. Now, of course, the consequences of that are very different, because if that desired allele is then inherited by offspring, it's going to affect all the tissues of those offspring. And I think we can say it will affect all tissues of all future offspring, because it's going to be passed down the generations. So how is gene therapy done? Now, as I've mentioned already, it uses vectors to deliver the new allele into the cells. Now, depending on your syllabus, you might need to know a little bit extra about the vectors that can be used. And there are two types that most specifications are going to want you to be aware of at least. The first one is viruses. Now, viruses, their whole raison d'etre is to inject their DNA into cells so that they can replicate. So it almost seems obvious that we use viruses as vectors. They're almost designed, dare I say, to do the job that we want them to do. Now, notice, though, that we genetically modify those viruses to make them both non-pathogenic and to contain the desired allele in the form so that that desired allele can be integrated into the host cell's DNA. And therefore, it's used to deliver, now if we're talking about those examples of muscular dystrophy and SCID, it's used to deliver dominant alleles into the blood stem cells of SCID patients and into the muscles of muscular dystrophy patients. The other form that you might need to be aware of are liposomes. Now, these are little spheres that are made up of a phospholipid bilayer. And that phospholipid sphere, as it were, contains the desired allele. And as a result of them being very tiny, and here's the important thing, they are really tiny, they are able to pass through both the cell surface membrane and the nuclear membrane to deliver that desired allele into the nucleus, deliver it to the host cell's DNA so that it can be incorporated into it. And this is the type of vector that is used to deliver the CTFR gene, the desired, the functioning, as it were, or maybe I should say, it delivers the CFTR allele that produces a functioning chloride channel into the lungs of cystic fibrosis patients. And that means that they can relieve their symptoms because now the lining of their trachea and their bronchi and their bronchioles has the functioning CFTR allele in them. And therefore, those cells make a functioning chloride ion channel. Now, interestingly, for cystic fibrosis patients, the lining of our trachea and our bronchioles is replaced about every 2 weeks, just by the way that those cells naturally replicate. And as a result, the gene therapy also has to be repeated every couple of weeks or so, because of course, the cells are being reproduced. Now, advantages and disadvantages of gene therapy. Now, there's a really obvious advantage to gene therapy, is that it provides treatments for diseases which up till now have had no previous treatments. Oh, I've just spotted a question. Do we need to know some examples of autosomal and X-linked diseases? No, you don't need to know particular examples. It's certainly not for AQA and OCRA. But however, there are classic examples that are always talked about in that inheritance topic. So sex-linked, I'm thinking of sex-linked, a classic sex-linked disease that you have probably used as an example is haemophilia. So the disease that stops blood clotting. You wouldn't be expected to know details of particular examples. You'd be expected to be aware that they happen so that if in an exam question they talk about a particular sex-linked genetic disorder, you can apply your knowledge of sex-linked inheritance to the disorder that you're talking about. I've included the information here just so that you can make links between topics, not because you're expected to learn that detail. I hope that clarifies that. So here's the huge advantage of gene therapy, but there are disadvantages. Now, number one, it can cause an immune response. Now, this is often with regards to when viral vectors are used, because in effect, you are introducing a virus into the body. You are infecting the body with a virus. And therefore, number one, it can cause an immune or an inflammatory response. And dare I say, almost more importantly, possibly more importantly, the patient can become immune to that virus because, of course, their body's immune system has been able to fight it successfully and has those memory cells in the blood waiting for a second infection. So the patient can become immune. So that means that the patient can only be treated once with gene therapy, and you kind of hope that it works. Another issue with viral vectors is that they can cause cancer. And there's a particular example, which I don't think you'd be expected to know about in a SCID. I think that's OCRB students. But leukemia has been found to be caused because a viral vector caused the healthy allele to be put into the DNA of the cell by an oncogene, a gene that actually causes cancer. This would be a mutated gene, and therefore it actually ended up causing cancer in SCID patients. In terms of germline therapy, that therapy that causes the alleles in sperm and eggs to be changed, of course, huge advantage is that you can cure a genetic disorder permanently throughout generations, potentially. The downside is that this is causing an absolutely irreversible change in the human genome. And it also means that actually the descendants, now I know this might sound a little bit weird, but they've had a treatment for which they haven't even been told about and that they haven't been able to give consent for. And giving consent for something is one of those fundamental ethical issues. Any patient has to give consent for the treatment, having been explained the pros and cons. Now, as a result of these ethical considerations, germline therapy is not currently allowed. And I think we have pretty much come to the end of my little live stream, my goodness, 20 odd minutes on gene therapy, which has covered certainly pretty much a lesson's worth of material. So I'm hoping that that has been useful. So I'm going to summarize what I've talked about, but if you have any questions, do start typing them up now while I do a quick summary. And if we're all done and finished, then I will let you go off and have a coffee. So we've talked about the fundamental use of gene therapy. We've talked about how it works in terms of how vectors are used. We've talked about particular diseases that EDUCAS and OCRB students need to know about. And we've talked about the advantages and disadvantages, some of those ethical considerations for this topic. So I hope that has been helpful and short and sweet. So unless there are any questions, I'm going to let us all go off and have a nice short lesson this afternoon. And next week, oh, here am I thinking, what have I got in the poll for next week? Oh, I think we're doing monoclonal antibodies next week. That certainly seems to be winning in the poll on my community tab. Do have a look at the community tab if you want to vote for next week's lesson. And if you are interested, if you find the way that I explain things useful, do have a look at my website, www.biologycarol.co.uk. You'll find all of my videos there. That website is my effort to make A-level biology accessible to every student, regardless of their family income. Okay, guys, I'm going to let you go. Lovely to see you and see you next Tuesday. Bye-bye.